Hi, I'm Kavelle Hylton, Chief STEM Ambassador for STEMBuilders Learning Hub, an online learning platform that is dedicated to STEM skill development. And today I want to assist you with your K biology exams. So I'll be looking at some of the ingredients for examination success. So I know you are all worried right now, especially during this COVID-19 pandemic. And so we're here to help. So I've been a tutor for many years. And so just like and subscribe to our channel and stay tuned. We have a whole lot more to share. So let's go. Right. So I'll be looking at achieving excellent grades in the K biology exam. So whether you're doing Unit 1 or Unit 2, you can do very well. I have students that have achieved 100% pass rate over the years. And so I have a bit of experience with this, right? So let's go. So I'm going to look at what they didn't tell you about the K biology exam, as well as some ingredients for exam success. So in 2018, 83% of the students that sat K biology Unit 1 exam achieved grades 1 to 4. So that's 83%. While 92% of the students that sat K biology Unit 2 exam achieved grades 1 to 4. And you find that students tend to feel that Unit 2 tends to be a bit easier than Unit 1. I do believe that might be true, or it's just a matter of students settling down by the time they get to upper 6. They're now ready, they have already passed the CSEC phase and they are more settled. So students tend to do well in Unit 2. So only 17 and 8% respectively of the students that sat the K biology Unit 1 and 2 exams in 2018 failed. So a very small percentage. So what are the ingredients for exam success? What do you need to add? What do you need to add to be successful in the K biology exam? So let's look at that. So one of the things that you definitely need is to be able to understand basic concepts. No, sometimes we think the concepts are not so basic because sometimes students don't grasp them as well as they should. And one of the things that I do when I'm teaching is to ensure that the basic concepts are clearly understood. So this is essential at this level. Basic concepts must be clearly identified and adequately revised. So according to the K biology examination reports 2006 to 2013, factual knowledge is important, but such knowledge cannot be readily applied if there is a little or no understanding of basic concepts. So understanding of a monohybrid and a dihybrid test cross, or the ability or to apply a chi-square test to a given data. So if you're doing Unit 1, this is extremely important because genetics, as you know, sometimes is seen as a problematic area for students, but they must be able to apply a chi-square test to a given data, as well as they must understand the difference between a null hypothesis and an alternative hypothesis, as well as the concept of probability. Understanding of the concept that translation is an RNA-directed process. So it uses codons to translate into amino acids, and they must have the ability to decipher a codon, right? So you must be able to read it. You must be able to understand that that is the process of translation. So given that information, you should be able to interpret it. So understanding also the difference between a phenotypic ratio and a genotypic ratio. Now, this is something that I see all the time. So when I would give my students to do a genetic cross, and you ask them, what is the genotypic ratio? And they'll readily will put that 1 to 1 ratio for a monohybrid cross. But then when you say, what is the phenotypic ratio? Sometimes students are not so clear on that. So definitely having a basic understanding of the difference between a phenotypic ratio and a genotypic ratio and what they generally are for both monohybrid and dihybrid inheritance, as well as understanding and specifying the categories. So for phenotypic ratio, in terms of the categories, then you would now look at, say, coat color. So 3 black and 1 white. But for the genotypic ratio now, categories, you need to be more specific to see how many heterozygotes or homozygous, dominant or recessive. All right, so the concept of macromolecules. Now this is basic in terms of polymers. Even at the CSEC level, you should have a basic understanding that monomers come together and many monomers make polymers, right? So I like to say monosaccharide plus monosaccharide equal disaccharide. And if you add more disaccharides or add more monosaccharides to a disaccharide, you'll definitely get a polysaccharide. Example, starch, glycogen. And the same principle applies when you look at protein in terms of amino acids combining to form peptides, dipeptides, and polypeptides. So understanding VO2 max, but this is for Unit 2, understanding VO2 max as being a maximum rate of oxygen consumption attainable during maximal or exhaustive exercise and continuing increase in exercise intensity does not result in an increased rate of oxygen consumption. Because of course, you know, you will definitely have anaerobic respiration taking place. So even though you have an increase in exercise intensity, does not mean that you're able to use more oxygen, right? Because you're maximum rate of oxygen capacity can't go beyond that. Understanding the difference between systolic and diastolic blood pressure. For example, describing systolic blood pressure as high blood pressure instead of maximum pressure on the arterial wall is incorrect. So don't describe it as high blood pressure, but rather maximum pressure on the arterial wall. And diastolic blood pressure should be described as a minimum pressure on the arterial wall instead of low blood pressure. So these are extremely important concepts that you have to understand. Understanding that ADH regulates the function of the collecting duct. That is, ADH targets the membrane of the duct to make it more permeable, thereby allowing water molecules to be reabsorbed. Understanding the concept of homeostasis, describing it as maintenance of an internal steady state in living organisms. And of course, you remember from the CSEC level, remember homeo meaning the same, right? So keeping it steady, keeping it the same without any changes, right? Or even if there is a change from that steady state, you're able to, the homeostatic system is able to bring it back to that set point. So that is a receptor detects, so understand the concept of homeostatic system. That is, a receptor detects changes in an internal environment, that a set point is the optimum condition. So at the CSEC level, you would have remembered this as being the norm or the normal point, right? So it's an optimum condition. So you realize at this level, you definitely have to use the correct terminology. The ingredients for exam success continued, right? So be competent in the use of technical terms, as I just mentioned. So examiners definitely look for keywords and technical terms in your responses. So even when we mentioned earlier, optimum condition, you know, we talked about, instead of saying low blood pressure, or we say minimum pressure on the arterial wall. So very specific there, and ensuring use of technical terms. So in the definition of evolution, terms such as slow in time, gradual change through successive generations, genotype, survival, and natural selection. So if you're doing Unit 1, when you're looking at evolution, those are terms that you can use. Also for Unit 1 students, try to stay away from describing organelles or cells as being small or large, as very, I wouldn't even say CSEC, but lower school level. So for sizes of cells, ensure that you use measurements and units. So between 0.5 to 10 micrometers and etc. Be able to interpret information presented in a diagram or in graphical form. Extremely important. Right, so here we have interpreting diagrams and graphs. Now, this is when the examiner will basically assess your practical skills, right? So we'll be looking at your experimental skills in this part of the question. It's usually the beginning of the paper as well. So for Unit 1, you're looking at analysis of specific nutrients in common foods, and you're using known reagents. So definitely food test is one of the experiments that you definitely have to know. You have to know what you get in terms of your conclusion or your results, and you definitely need to be able to identify the food substance and the food test reagent that goes with that. So here we have state the nutrient being tested for by each of the following tests. So we know that that would be protein, Benedict solution, reducing sugar, not just sugar, just reducing sugar. Lugol's iodine, you're talking about starch. Grease spot test, you're talking about lipids or fats and oils, right? And you will get half mark for each, so it's a total of 2 marks. Using the data presented in Table 1, you determine 2 food substances which contain all 4 nutrients being tested for in this investigation. All right, so using the data presented, 2 food substances which contain all 4 nutrients being tested for. So if you look at eggs, Urate solution purple, Benedict solution blue, iodine black, but the grease spot none seen. So this contains everything. Well, it doesn't contain sugar, contains protein and starch, but no fat, no sugar. Milk, pink, so there's some protein there, but the intensity or the amount is smaller amount, so you'll realize that intensity of the color would differ. So pale yellow, little dark blue, but no fat. Cheerio cereal, pale blue, brick red, blue black, only seen when food sample is dissolved in alcohol, okay. Hamburger patty, purple, blue, yellow, so not changing either. Carrot, orange, dark blue, none seen. Potato chips, pink, brick red, black, okay. Pepperoni pizza, donuts. I'm seeing donuts here, pink, orange, dark blue, clearly seen when rubbed on the paper. So we're looking at pepperoni pizza and donut as being the answers for that question. So it's 2 food substances. So for the 2, you get 1 mark. All right, so with reference to the qualitative results in Table 1, suggest which foods are good sources of nutrients for cell structure and growth of the cell and for supply of energy. Justify your answer with a brief explanation. So for cell structure and growth of the cell, which food substances can we look at? For cell growth, you're definitely looking at, for growth, you're definitely looking at something with protein, and for structure, you're looking at something with the lipids, right? So eggs, hamburger, patty, and pepperoni pizza are good sources of protein, and donut is a good source of lipids. Both nutrients are very important for supporting cell growth and structure. With respect to a key nutrient for providing energy, all food items with good starch content could have been listed. Also, you definitely need to mention that a strong positive color change as justification for the selection of the food item. So it asks, to justify your answer with a brief explanation. So it's 2 marks for each part of the question. If you're doing Unit 2, then you look at photosynthesis, the rate of photosynthesis. So see, light intensity on rate of photosynthesis. Now students always seem to confuse this graph with enzyme graph. And so it's important that you ensure that you look at what is on the X and Y axis and that you focus. You're looking at photosynthesis now. So effect of light intensity on the rate of photosynthesis, you're going to use double-headed arrows on the graph to identify the regions where light is a limiting factor as well as other factors become limiting. And you're going to look at what is the significance of each point, D and E, on the X-axis and Y-axis. So point D, this represents optimum light intensity. That's what it is. So it's optimum light intensity. You realize that the graph levels off after that, which is saying to you that another factor becomes limiting. So that's E. So this represents the maximum rate of photosynthesis. Photosynthesis cannot go beyond this unless you increase the other factors. So here, light is a limiting factor because the light intensity is low. And here it is when other factors become limiting. Light intensity is increasing here, but it still levels off because carbon dioxide or something else has become limiting, right? CO2 levels more than likely. So in a greenhouse, you tend to find that it will increase the CO2 level and more photosynthesis will take place. And then of course, they would also have an artificial light source. So the light intensity would just continue to increase as well. Understand Rubik terms, for example, comment versus discuss, or outline versus explain. And this is critical in the quality of your response. If you don't understand the Rubik term, then you won't know how much to write or what to write. So you might just write everything that you know, but you still did not answer the question. So comment means to state an opinion or view with supporting reasons, while discuss means to present a reasoned argument. You consider both points, for and against, and explain the relative merits of a case. And in the situation where you are writing an essay question and it's asking you to comment or to discuss, ensure that you plan your responses. So example, discuss two significant differences with respect to the timing of oogenesis and spermatogenesis. Response 1. Oogenesis takes place once every month, while spermatogenesis can take place every day. Is this correct? Right, so two significant differences. Have they explained two? No. Discuss means to present arguments for and against, right? Let's go back to that. Reasoned argument, consider points both for and against, and explain the relative merits. So this would have been a poor response. A correct response would be females have the full complement of a fixed number of primary oocytes from birth and produce ova from them between menarche and menopause. While in males, so you see, we give in one side what's happening with the females, and then move to what's happening with males. So while in males, primary spermatocytes develop from the germinal epithelium. So you are seeing the difference. Fixed number of primary oocytes from birth, spermatocytes developing from germinal epithelium. And it takes place when? After puberty, and continue throughout life. Further, whereas the development of mature sperm takes approximately 70 days, oogenesis can take as long as 48 to 50 years. The formation of the mature sperm from spermatocytes is smoothly sequential, whereas in oogenesis, the final mitotic division does not take place from many. So you definitely see the debate here, the comparison, what's happening with this, and I like how this is presented, because, and sometimes what persons will do is write everything that happens for oogenesis and then write everything for spermatogenesis. But it's always good to show the comparison, and many times when I'm teaching this topic, perhaps a table is what is used, and you're comparing point for point. And so this is nicely done and would be a very good response. Outline means to give basic steps only. So example, outline 3 steps in the method for producing human insulin commercially. And explain means to give reasons based on recall. To explain how human insulin is produced. So here you're going to outline just the 3 steps, and you're looking at the method for producing human insulin. So step 1, step 2, and therefore. While in this case, you're going to now explain how it is produced. So it's way more detailed, and you're going to give reasons as well. These are some other Rubik terms that you can use. Not that you can use, that is often used. These D words, especially deduce, define, describe, determine, ensure that you know what they mean so that you can answer the question correctly. So analyze, explain, identify, interpret, list, name, state, right? Another key ingredient, which is the final one, for examination success is to ensure that you have excellent test-taking skills. And that's one of the main reasons students fail their exam, right? So there are some test-taking do's and don'ts. So watch our next video and we will definitely be going through them step by step. So there's more to taking an examination than just memorizing the content. So yay, you know, you have a basic understanding, or you have an understanding of the basic concepts. You're clear, but can you take a test? So watch out for next video and keep calm and pass your K-Biology exam. I'm Kavelle Hylton, Chief STEM Ambassador for STEMBuilders Learning Hub. Remember to like and subscribe to our channel. Thank you.